In this video, we'll take a look at some of the staining procedures that are at the foundation of the microbiology lab. The slide that we have on the screen here really is meant to emphasize what goes into preparing most specimen for light microscopy. There's two major ways that we try to visualize or try to prepare those specimens. You can utilize something called a wet mount. And a wet mount is simply when you place your specimen within a drop of water, and typically you lay a cover slip on top. The drawback to that wet mount obviously is that since you have your specimen typically in a drop of water, and the water being colorless and the specimen also being colorless, it can be hard to visualize. Once again, we typically stain organisms to improve that contrast. And in many cases, not all, but many, a staining procedure will begin with performing what's called a smear, or making what's called a heat-fixed smear. As you see at the bottom, these are the steps in making a heat-fixed smear. You'll start with your specimen. You'll add a small amount of liquid to the surface, typically water or some type of serum. You'll aseptically remove your specimen from your sample, and then you'll mix it into that drop of liquid. You want to smear it or spread it across the surface of your slide. Following the organism being smeared across the surface, you'll allow the mixture to air dry. After you air dry, you'll pass it through the flame of a Bunsen burner quickly, roughly about 3 times. This is called heat fixation. The heat fixation slightly denatures proteins found in the membrane of the organism. That denaturization allows for the organism to stick or become fixed to the slide, so that as you add different reagents, your organism won't be washed away. Certain stains only require one dye. We'll talk about those in a moment. But after you've heat fixed your smear, you can apply the stain or the reagent that you're using, and then stain your slide, or your cells, and then observe under a microscope. Table 3.2 gives you a summary of various staining techniques that you'll see performed within a microbiological lab. You've got your simple stains. Your simple stains only use one dye. There's a corresponding stain also called a negative stain, which we'll take a look at as well. Differential stains allow you to tell the difference between organisms. I'll talk extensively about the gram stain and a little bit about the acid-fast stain. Special stains are used to stain specific structures. You've got capsular stains, which are used to stain capsules. Endospore stains, which are used to stain endospores. And flagella stains are used to stain flagella. And then you have your fluorescent dyes and tags. The fluorescent dyes typically attach to various molecules within an organism, and once placed under ultraviolet light, they'll cause the organisms to fluoresce. The fluorescent tags usually are some sort of fluorescent labeled antibody that will bind to a specific component, and once that is bound to that component, you can actually cause the illumination of the organism if it's placed under UV light. As I said before, a simple stain uses only one dye. Simple stains rather use only one dye. In order to accomplish a simple stain, a category of dyes called basic dyes are used. These basic dyes carry an overall positive charge. The reason that they're utilized is because the cells, most bacterial cells at least, themselves have a negative charge. So the old saying, opposites attract, still applies. The negatively charged cell attracts the positively charged dye, and ultimately the organism ends up being stained. Examples of basic dyes include stains such as methylene blue and also crystal violet. Acidic dyes can be used for negative staining. Acidic dyes have the exact opposite charge of basic dyes. Acidic dyes are negatively charged. So, unlike the basic dyes, the acidic dyes actually become repelled. Remember, light charges repel. So, negative dye, negative organism, the organism repels the dye, and instead the background becomes colored or stained, while the organism is going to be left clear or colorless. At the end of the day, the organism being clear or colorless with the background being stained gives you, once again, better contrast, which is our goal. So you absolutely want to keep in mind the two categories of dyes, basic versus acidic. Keep in mind the charge associated with each, positive or negative. And then keep in mind what staining procedure they can be used with. Simple staining uses basic dyes, negative stains use acidic dyes. And then as well, you'll want to keep in mind some of the examples of the basic dyes. Differential staining allows you to tell the difference between organisms. Usually, differential stains require you to perform or use a series of reagents. The most common differential stain performed in the microbiology lab is a gram stain, and we're going to use that. The gram stain takes advantage of the cell wall differences between most bacteria. Now, in order to perform a gram stain, there are 4 critical steps. Well, let's say 5 technically, because first you'd have to make a heat-fixed smear. After you make your heat-fixed smear, you'll apply what's called the primary stain. The primary stain is going to be crystal violet. You'll apply it for 60 seconds, rinse it with water, and then after application of crystal violet, all cells should appear purple. Next, we come back and we add the mordant. The mordant is actually a chemical called iodine. The iodine complexes with the crystal violet within the cells. Since we really haven't done anything to remove that crystal violet, all cells remain purple after application of the mordant. The third step is to apply the decolorizer. As its name implies, it's intended to remove color. The decolorizer is alcohol, specifically ethanol. What will happen is that the iodine, which is complex with the crystal violet, resists removal due to the decolorizer. However, in gram-negative cells, that iodine crystal violet complex can be easily removed. So the gram-positive cells remain purple due to their crystal violet iodine complexes remaining trapped, while the gram-negative cells, since the crystal violet iodine complex can be stripped away, will ultimately become colorless. Now, the reason that you can remove that crystal violet iodine complex from the gram-negative and not the gram-positive honestly relates to their cell wall structure, and we'll discuss that in a later video for chapter 3. Now we have purple gram-positive cells and colorless gram-negative cells. So let's try and improve the contrast with the gram-negative cells. We're going to use a counter stain called safranin. Safranin is applied to your cells for 60 seconds, and then rinsed away. Since the gram-positive cells are already full with the crystal violet and iodine complex, they remain purple. But since the gram-negative cells were now exposed and could be stained again, they should be red to pink in color. So at the end of the gram stain process, gram-positive cells will be purple, and gram-negative cells will be pink to red. Here you can see an actual photomicrograph of that end result. Notice the pink gram-negative organisms, and then you've got several clusters of purple gram-positive organisms. For the gram stain, I do want you all to keep in mind the reagents, what they are, or what their nicknames are, so to speak. So you should know, for instance, crystal violet is the primary stain. I definitely want you to know the appearance of the cells after application of each reagent as well. The acid-fast stain is another differential stain and is used to try to detect organisms from a specific genus called Mycobacterium. These Mycobacterium have unique structures within their cell walls called mycolic acids. It makes their cell walls very waxy. It also leads to the fact that they ultimately cause some really problematic diseases such as leprosy and tuberculosis. The acid-fast stain, is a multi-step process. I'm not going to give you all of those steps. If you have to perform it in lab, you'll get it there. But I do want you to appreciate what genus we're trying to observe with the acid-fast stain, which would be the Mycobacterium. We would say that the Mycobacterium are acid-fast positive, and most other genera are going to be acid-fast negative. The capsule stain is a special structure stain. I mentioned this to you earlier. You can see the organism here in the center, but notice the clear halo around the organisms. That would be due to the presence of a capsule. Endospore staining, another special structure stain. The endospore stain allows you to visualize endospores typically produced by 2 genera of bacteria, the Bacillus and also Clostridium species. The endospores are highly resistant structures that are essentially meant to allow an organism to survive harsh environments. The endospores at the end of that procedure typically appear green, so you see multiple endospores exposed throughout this entire field of view. But if you look really closely right in here, you'll see some endospores still within the cells. You see the green endospores, and you see the redness within the image actually is coming due to the cells being stained slightly red with the safranin that we mentioned before. So you don't have to know the steps in an endospore stain, but you should appreciate that the endospore stain is a special structure stain, and also that it ultimately is used to try to detect those endospores in both Bacillus and Clostridium species. The flagella stain is up next. The flagella stain is used to try and stain flagella, which are in most cases going to be used for motility by bacteria. Multiple arrangements for flagella exist. You can see here in this little organism, this would be called a peritrichous arrangement. The peritrichous arrangement allows the flagella to surround the entire perimeter, so to speak, of that cell. If you see, there's one flagellum here, another one. So there's multiple flagella around the entire perimeter. Polar flagella are those that occur at one end. Another term for polar flagella would be monotrichous. You can see all you got to do is take off the peri at the beginning and put M-O-N-O. Monotrichous means one flagellum. There's a term called amphitrichous. It's not actually on this image, but it does relate once again to flagellar arrangement. Amphitrichous is when you have one flagellum at each or at opposite poles. So it would be an organism that has two total flagella, but has one flagellum at one end and the other flagellum has to be at the opposite end. And then there's also a term called lophotrichous. That's L-O-P as in pot, H-O, trichous. Lophotrichous are when the flagella typically lump together, so to speak. So they will all be kind of grouped together in a tuft. Having talked a little bit about the basic staining procedures, let me give you guys some of this shape or morphology terms related to prokaryotic cells along with the arrangement terms. You see two basic morphology terms here. Coccus for any organisms that are spherical. Bacillus will relate to organisms that are rod-like in shape. Short rods are sometimes referred to as coccobacilli, or coccobacillus is the individual term. Rods can occur in different shapes as well, so they don't just have to be a straight rod. If it's a curved rod, it's referred to as a vibrio. You see that here. The spirillum is a rod, but it's in a helical formation, and the spirillum is a rigid helix. The spirochete would also be helical, but it's going to be flexible. And then some organisms vary their shape, and if they do have variable shape, we refer to those organisms as pleomorphic. As the cells divide, sometimes they produce classic arrangements. The most classic arrangement terms are strepto and also staphylo. Strepto refers to organisms arranged in a chain-like formation. So as you see over here, you have streptococci. Staphylo refers to organisms arranged in grape-like clusters. So you could also have staphylococci. Diplo is a term when two organisms are paired together. So you see here a diplococcus. You could also have diplobacilli. And then sarcina is when those organisms are paired, or rather arranged, I should say, into a cube-like formation. So you can have the cube-like formation of sarcina.